Welcome, everyone. We are so glad that you're here with us tonight for a very special post-symposium community webinar hosted by emilys-entourage. For those of you that don't know me, I am Emily Kramer-Gollenkoff, and I'm a co-founder of emilys-entourage. emilys-entourage is a 501c3 that speeds research and therapeutic development for those in the final 10% of the CF community that don't benefit from existing CFTR modulators. I'm also a person with advanced stage CF myself. We started emilys-entourage back in 2011 to speed life-saving advances for those in the final 10% of the CF community. We believe that every single person with CF deserves the chance to live a long, healthy life, no matter how rare your mutation. And so we race every single day to make that dream a reality and to make it happen fast, because we know that with a disease like CF, days matter. And so we've come a long way since those early days. Since 2011, we have raised over $18.5 million. We've awarded 42 research grants and secured over $52.3 million in follow-on funding. We deployed a venture philanthropy model to spin out a clinical stage CF gene therapy company, and we've advanced multiple other projects to clinical stage. And we've developed a global patient database called CF Clinical Trial Connect, or the CTC, and a clinical trial matchmaking program to accelerate clinical trial recruitment for this ultra-rare population. And really, every single thing that we do is singularly focused on accelerating research and drug development for people with CF that don't benefit from existing modulators. And so earlier this year, we gathered over 100 of the world's most preeminent researchers, clinicians, and industry leaders for the 2025 emilys-entourage Scientific Symposium, The Delivery of Genes, Advancements and Challenges Through a CF Lens. And so tonight, our goal is to share the most exciting insights and updates from the symposium with you, and give you an opportunity to ask your questions of our panel of expert CF clinicians and scientists. So the plan is pretty simple. We're planning to start with an overview of key takeaways from the symposium, which you'll hear from our esteemed CF clinicians and scientists. And then we're going to really spend most of the time answering your questions, those that were pre-submitted, as well as any that you want to ask live on the chat tonight. So our main goal is just we want to address what you are wondering about and what matters to you, and to provide you direct access to people who know best. And so please don't be shy, because we're here to try to get you answers. And so Dr. Chandra Ghosh, EE's Chief Scientific Officer and the mastermind behind our whole symposium, will be moderating tonight's conversation, and I'll pass it over to Chandra in just a moment. But first, I just want to say a heartfelt thank you to all of our panelists who have so generously agreed to be part of this webinar, to our amazing team at emilys-entourage for pulling this together so beautifully, and to all of you for being with us tonight. We are so honored and so grateful. And with that, I will pass it over to you, Chandra.
Hi, everyone. Thank you so much, Emily. Good evening. I'm so excited to be here today to share with you key insights from our symposium that we held a few months ago in Philadelphia. As Emily mentioned, more than 100 scientists and physicians were gathered in Philadelphia to discuss the topic of the delivery of gene therapy, which in our field is the Holy Grail. Let me share a few things that were really exciting about that happened during the symposium. One, obviously, was that it was really exciting to see so many scientists working together towards a cure or even a treatment, especially for those in the final 10% who do not benefit from currently approved modulators. The two most exciting aspects that I found to be really thrilling as a scientist myself were we were witnessing unprecedented advances in rewriting the genetic code itself. Scientists presented major progress in prime editing and CRISPR-based tools that can repair the defective CFTR genes at the DNA level. Perhaps most remarkable was the discussion on fetal therapy, where we could imagine a future where children with CF are born free from the burden of this disease. We are no longer in the realm of science fiction. This is a reality in many other diseases. Second, as an infectious disease scientist myself, it was really interesting to look at the science behind how we are tackling the infection crisis with new weapons. Researchers introduced silver nanoparticles that eliminated deadly bacteria like Pseudomonas and staph without creating resistance. CRISPR-engineered bacteriophages acted as precision missiles, breaking down biofilms that protect harmful bacteria, and DNA-encoded antibodies that were neutralizing infections before they even started creating any harm in the lungs. What made this moment most significant is the science and the collaboration. By bringing together leaders from academia, biotech, and pharma, we were addressing complex challenges that required collective expertise. For members of a community who have waited for so long for effective treatments, these advances represented meaningful progress. They pointed towards potential therapeutic improvement beyond current symptom management approaches. So a few housekeeping items before we start. As you have noticed, the program is being recorded. Attendees will not be recorded in any way, but the panelist presentation will be recorded and Q&As will be captured to continue to share information beyond the event tonight and to inform future discussions. Closed captioning has been turned on for tonight's event. Feel free to activate it on your screen if you'd like to see the captions. To ask a question during the Q&A portion, enter your questions into the Q&A box and then click send. We will do our best to answer all the questions as time allows, but we may not get to all of them. And we ask you to hold live question till closer to the end. If we don't answer, we aim to do so in a recap blog post after event. Also, we are not able to answer any medical questions. So for medical advice, please contact your care team. Please complete a brief survey at the end of the presentation. Feedback has been so helpful for us, and we want to make these events better. Please wait 3 seconds after the conclusion of the webinar before closing the browser window in order to see the pop-up survey. Zoom will automatically launch the survey in your default browser, even if you're using a different browser to view this webinar. Lastly, a brief legal note. The speakers as well as EE may have financial relationships with commercial interests, including support for campaigns, events, recruitments, and programs. These relationships do not affect our opinions at all, which are shared this evening, or signify any endorsement of any specific trials, drugs, or companies. Tonight's event is not sponsored by any outside company. So without further ado, let's get started. We are so fortunate to be joined with 3 brilliant minds who have long been involved in CF research and who have attended this past symposium. I'll ask them to briefly introduce themselves. We will start with Dr. Jen Taylor-Kauser.
Hi, my name is Jen Taylor-Kauser. I'm an adult and pediatric pulmonologist at National Jewish Health in Denver, where I lead the research program in CF.
Next, we have Patrick Harrison.
Hi, everyone. My name is Patrick Harrison. I work at Cincinnati Children's. I've been here about 18 months. Prior to that, I was in Ireland for 20 years, where we've been doing gene editing for CF since about 2006.
Thank you. And lastly, we have Dr. Paul McRae.
Hi there. I'm at the University of Iowa. I'm a pediatric pulmonologist, and I have a long-standing interest in CF lung disease, how it gets started, how to treat it, and especially genetic therapies.
Thank you so much.
Pleasure to be here.
Thank you, Paul. Without further ado, I will pass it on to Dr. Harrison to get started.
Thanks, Chandra. If you can. So I've got a few minutes to talk about gene therapy. The first clinical trial for gene therapy was performed in 1993 in Iowa. I don't have a photo of that one, but this was a second trial performed in Philadelphia, New York, a year later. Next slide. But if you can do the next slide, please. But if you go on 30 years since those early clinical trials, we've now got 12 drugs which have been approved for gene therapy. But if you look closely, you won't see the name of a drug for cystic fibrosis. If you look in the list of diseases, you won't see cystic fibrosis. So why is that? Next slide. Let's just briefly explain the problem here. In 1989, when the gene was cloned, we found out the sequence of the genetic material, the normal CFTR gene. Next. This gives rise to a messenger RNA. That messenger RNA then encodes a protein. Next. And that protein traffics to the cell surface, opens as an ion channel. Next. And chloride and bicarbonate ions come out and our lungs work normally. The other thing that was discovered was this mutation, which many of you have heard of, F508del. This variant means the protein doesn't traffic properly. But, as we've heard, most people with CF can benefit from modulators. Next. And these modulators help this protein move to the cell surface and open, and we know the massive benefits that this can give. But the other thing that we discovered a few years later was there's lots of different mutations, including this one, G542X, other mutations, 1282X. You've all got mutations you're aware of. These mutations don't respond to modulators, but in theory, they could work with a cDNA therapy. That was shown in the lab just 1 year after the gene was cloned 30 years ago. The cDNA is a small copy of the gene that makes an RNA molecule, which makes the protein, and that can traffic. This gives us the basis for so-called cDNA therapy. But over the last couple of years, as RNA technology has advanced, next slide, we can now have an option for mRNA therapy. Next slide. So what are the options? There've been a lot of clinical trials, but as you'll see on the next slide, there's at least 7 which are currently ongoing. Dr. Taylor-Cowser will talk more about this later, but the point I want to make from this slide is that we've got all these different opportunities, messenger RNA, cDNA, lots of delivery options, which Dr. McRae will talk about when I finish. But what if these don't work quite as well as we hope? Or what if their effects don't last quite as long as we would like? Well, this is why we're interested in gene editing on the next slide. Gene editing is often drawn this way as a surgeon using a pair of scissors, but everything's operating on a much smaller scale. And gene editing will really benefit from other additional insights that we get from these clinical trials, which you'll hear more about a bit later. Because we're not delivering a pair of scissors or a scalpel, we're delivering a small protein known as CRISPR-Cas9. And that protein will be encoded by a cDNA. So we need to know how cDNA therapy works for CF. Or a messenger RNA, or Dr. McRae's lab have even delivered it as a protein in some model systems. And what that protein does, that little grey blob there in the middle of the slide, next, that protein can bury into the nucleus of the cell. It can scoot along and find mutations. And as shown on the next slide, it can correct those mutations. And if it does that, we make a new protein and potentially we can fix that cell. And a key thing is we can fix that cell permanently. Next slide, please. So we don't use a scalpel or a pair of scissors. We use this protein, and I'll briefly explain how it works. Next slide. I like to use my mini word processing keyboard to explain this. At the bottom there, we've got the sequence of the CFTR F508del mutation, just 3 nucleotides missing. So if we click through these slides, the first thing is to find the mutation. And that's what CRISPR does. It searches the entire human genome, 6 billion base pairs, and can find a very precise region in the cell. If it can find that region, in this case, the CFTR gene, it can cut out that region. And then if we introduce the correct sequence, which only needs to be very short, a so-called donor sequence, we can then copy the information which is missing. Next slide. And we can paste that material into the genome. And if we can do that, we can repair the cell. Now, this isn't a theory. This is an experiment which was done 12 years ago by Jeff Beekman's group. Next slide. And many of you will recognize this model system. These are the human intestinal organoids. On the left-hand side, you've got wild-type organoids, which swell. In the middle, we've got the F508del organoids, which don't swell. But on the right-hand side, they've not been treated with modulators. They've had the gene sequence corrected and they behave exactly as wild-type cells. But these are cells in the lab. How do we get these into the person? Well, if we look on the next slide, this has been done for other diseases. Many of you may have heard of Victoria Gray, who was the first person to be cured of sickle cell disease by gene editing. But in her case, she had her bone marrow cells removed, edited, and successfully put back. For other diseases, there's been a big trial of alpha-1 antitrypsin. This has also progressed in a phase 2 trial with very positive results, but the difference is we can deliver that to the liver. So on my final couple of slides, I just want to talk about some of the options which are available. We recently published a study, and what this graph shows is the more editing you get, the more CF function you can restore. And there's 27 studies published by groups all over the world in this area. If we look at the next slide, I'll just briefly explain them and finish on this slide. So, base editing seems to be the one to go for. It's been used in a clinical trial. It's been used to treat several different people, but we can only do one mutation at a time. And this is why we're interested in prime editing. Prime editing can work on small clusters of mutations. So if you developed a treatment based on that, you could treat more people, but it's not quite as efficient. What if you could correct all the mutations in a whole exon? There's techniques such as template jumping, which we heard about at the conference. But again, the efficiency is not quite as good. If you want to try and treat lots of people with the same drug, you'd need to correct mutations in lots of different exons at the same time so that all these people could benefit from the same treatment. But again, the efficiency goes down. But the editing efficiency is there, and that editing is permanent. If we click on the next slide, we can see all of these technologies can be delivered either by RNA or click, and the next slide, by a DNA technology. And this is what Dr. McRae will now introduce. Thank you.
Thank you for that introduction, Patrick. So we'll carry on here. One of the things that people always wonder about is what have we learned from past gene therapy trials? You're probably aware that there, especially in the 1990s and early 2000s, there were clinical trials with CF gene therapies. And to try to briefly summarize some of the outcomes of those studies, I think one of the take-homes was that the barriers to delivery of genetic cargoes to the lung were more formidable than people originally imagined. And that led to the realization that the efficiency of delivery was less than anticipated and maybe not enough to be therapeutically relevant. Those first studies were all done with delivering DNA cargoes to make the CFTR protein like Patrick was just describing. Also, when there was correction observed, it was generally short-lived. And it could be that immune responses to the vector, to the delivery systems played a role to that. And in the subsequent years, people have learned a lot more about the delivery vectors that I'll mention in more detail in a couple seconds. And also realized the importance of using models that were rigorous and stringent. It's very easy to deliver these kind of cargoes to simple cell models, but when you move to more complex multicellular tissue models or to animals, it's much more demanding, and we've learned that the things that work in these more stringent models are more likely to translate to being effective in people. You could, next slide, please. So, just talking about the difficulties a little more, think about your lungs. We think they're inside our body, but they're really exposed to the outside, and it's a large surface area that's really evolutionarily designed to keep foreign materials out, including the therapies that we're trying to deliver. There's a multi-layered defenses, inhaled or delivered gene therapy particles could be trapped in mucus and swept out of the lung. There are host defense molecules that can inactivate these vectors. And there may be inflammatory responses or immune responses that lead to elimination of the targeted cells. These are all things we have to think about in designing next generation therapies. Another thing we wonder about is, in a perfect world, we would love to have a delivery strategy that went to every organ affected by cystic fibrosis and correct things, as Patrick outlined. And that's our big picture goal. But the most practical short-term goal is to focus on the lung, because most of the morbidity associated with CF is related to the lung disease. And that's the focus that most people are trying to tackle right now. So unlike these blood diseases such as sickle cell, where you can take the cells outside the body, correct them, and put them back in, we do not have that luxury with CF. So most of the therapies that we're talking about for lung disease involve an inhaled form of a delivery vehicle. Next slide, please. Just to hammer home some of the complexities here, if you think about our lungs as sort of the way branches in a tree grow, there are 23 generations of progressively smaller airways with different cellular anatomy and diameter that also pose delivery challenges. A lot of the advances in gene therapy right now are happening with this ex vivo therapy, where you take the cells out and correct them, or if you happen to have a problem that's primarily related to a secreted protein or a liver disease, the liver seems to be very willing to have gene therapy vectors delivered to it. It's easier than the lung. So some of the complexities have to do with the architecture of the lung itself. And I also mentioned here some of the different cell types that we might want to target. So if we go to the next slide, really one of the questions is, where are we trying to get all this stuff to? And in a very simplified model here in this cartoon, the lining of the airways has multiple cell types. The most abundant ones in the large and small airways are the ciliated cells with the little hair-like projections that help move mucus along. The secretory cells that they produce proteins that are involved in host defense. They're also an abundant site for CFTR function. We think they're very important for CFTR function. And then there are some rare cell types, such as ionocytes, that may have a lot of CFTR, but there are very few of them compared to these other major cell types. And there are also mucin-producing cells like this green one in the middle. Now, if you go to the next slide, underneath that layer in the large airways are basal cells. And it turns out they're the progenitor cell or the stem cell that make all the surface cell types. So one of the things we think about a lot with genetic therapies and delivery is, if we can get something into these basal cells and correct the gene or permanently deliver the cDNA to make the protein, then when those red cells go on and make all the other colors of the rainbow, we will be able to correct those surface cells. And if we can permanently correct it in the basal cells, we would expect that we'd have a lasting therapy. Next slide. So we know those surface cells turn over. They probably have lives of many months. We don't exactly know in humans, but as they turn over, the basal cells make new ones to replace them. Next slide, please. Just to briefly touch on some of the delivery approaches, I've divided them into viral on the left and non-viral on the right. On the viral side, one of the potential advantages of viruses is that they've evolved over time to effectively get into our lungs. And so if we could harness some of that evolutionary strategy, that could be beneficial. So I've named a few of them there: adeno-associated virus, bocavirus, lentivirus, herpes simplex virus. On the right are non-viral vectors, which have emerged in importance more recently. And there's a lot of work going on several different types of particles, whether they're lipid derived or nanoparticles with other components. These are all under investigation, and also virus-like particles and peptide delivery approaches. Next slide. This is a little wonky here, but I meant to highlight the classes of vectors that are in clinical trials now, which we'll hear about shortly, but that includes adenovirus, lentivirus, herpes simplex virus, and lipid nanoparticles. Next slide, please. So as we've heard, we're learning a lot from advances in other genetic diseases, and we want to apply this knowledge towards CF. And the ongoing clinical trials for CF, the outcomes of those are going to influence our approaches, and we'll hear more about that from Jennifer. Next slide, please. I wanted to briefly touch on a couple of topics that I thought were particularly exciting. The first one relates to non-viral delivery with lipid nanoparticles. So there were several talks from industry and academia teams who were trying to advance forms of lipid nanoparticles to deliver RNA in particular, and also virus-like particles. Next slide, please. So one of these was from Marco Weinberg, who represented Recode Therapeutics, which is doing one of the CF clinical trials. And they're interested in both delivery to the lung and also advancing particles that might allow delivery systemically to multiple organs. So these are particles that have 5 or 6 different lipid components. They presented data showing that they were able to restore CFTR function in a robust human cell model. The trial is ongoing. They are also teaming up with Intellia, which is a gene editing company, to try to use this technology to deliver a base editor to the airways, and they're continuing to work on this systemic delivery approach. Next slide, please. There were also talks about viral vector delivery approaches. I wanted to highlight briefly one from a colleague at Iowa, Patrick Sin. He and Ashley Cooney here presented some results related to a way to formulate vectors that enhances their delivery. And this is a very simple thing where they found that hypertonic saline, which is an FDA-approved treatment for CF, when you formulate the vectors in hypertonic saline, it increases the access to the cells and improve the delivery of several classes of viral vectors that are 2 of which are in clinical trials. And this simple formulation approach may have broad applications for delivery. Next slide. So just briefly, I wanted to pose a few questions that we continue to think about. One is, if we can correct, can we deliver the goodies to the cell types of interest? That's a major question with each technology that we need to address. With each technology, we need to understand how long expression will persist, if correction of the surface cells will be sufficient to correct the defects in the airways. Will there be immune responses? This is baked into the trial design. And can we develop something that will work for organs other than the lung too? Next slide. So we continue to be informed by other genetic disease studies. And, we're optimistic about where things are headed, and we're committed to developing genetic therapies that work for all people with cystic fibrosis. And I'll stop there.
Thanks, Paul. That was a great setup for the next part of the discussion. So go ahead, Chandra. So back in 1989, when the CF gene was discovered by a collaboration between Lapchi Choi, Jack Riordan, Francis Collins, Batsheva Karem, and others, it really seemed like gene therapy would be around the corner. As you heard from Paul, that didn't actually happen because we weren't getting enough of the corrected information into the cells, and people were having immune responses to those different vectors, the way we get it into the cells. And so we had to go back to the drawing board. Next slide. So as a clinical investigator who is involved in designing many of these trials, these are sort of the things that I am thinking about, and they'll reiterate some of the things that Paul just said. So in the preclinical stage, before we start giving it to people, the question is, what is the best way to get it into the cells? So which vector, meaning, is it going to be the lipid nanoparticles, or is it DNA using a virus? We still haven't quite figured that out yet. We also, as Paul alluded to, aren't exactly sure which cells that we have to target. We think that we want to get it into basal cells because that's where it will last the longest, but we're still looking at that as well. Also, how many of the cells need to be actually successfully transduced, meaning we got the genetic material into those cells and we made the protein. Is it 20%? Is it 50%? Something else that we're still trying to determine. And then what measures of efficacy or how well it's working do we need to see in cell lines and in animal lines before we think it's ready to go into people? So if we go to this early phase study, so meaning phase 1 studies where we're mostly looking at safety, or phase 2 studies where we're looking at safety, but also about whether it's working or not, the question is, what measures of efficacy do we need to see there? So how do we tell if it's working? Is it enough to look at the cells and say, yes, we actually got the material where we wanted it and in the correct cells? Or do we actually have to see improvements in these very early trials in things like lung function? Also, as we've heard, inflammation has been a problem. And the question is, how much inflammation is okay, and what measures of inflammation do we need to use before we again say, well, this is doing well enough that we can move it into a much bigger trial? And those are the phase 3 clinical trials. And those trials are the ones we need to have regulators like the Food and Drug Administration or the EMA or the Canadian Drug Board review them and say, yes, it's ready for us to approve this drug. So again, we have to ask, what measures of efficacy do we need to use? So the Food and Drug Administration says that a drug needs to improve how someone feels, functions, and survives. So they have to live longer, if you can measure that, or they are functioning better, or they feel a lot better as measured by things like quality of life. But are those the right measures for these genetic therapy trials? We're not quite sure yet. Also, how much improvement is enough? So when ivacaftor or Kalydeco was approved, the lung function improvement was about 10%. But then when the next drug, lumacaftor, ivacaftor, or Cambi, was approved, it only improved lung function by about 2 to 4%. But that was enough to stabilize people, many people, to help them avoid transplant and or to wait until Trikafta was later approved. So is that enough improvement? That's under debate. Also, a topic that comes up frequently is which people with CF should be in these initial trials. Is it everyone who doesn't have access to modulators? What about the people who can't tolerate the modulators because they have side effects from them? Also, we have to think about lung function. We don't want to include somebody whose lung function is so low that we might put them at risk, but also if we include people whose lung function is very high, we might not be able to measure how much the therapy is improving the lung function. So those are some of the things that we're thinking about, though. There are also some really critical participant considerations. So historically, and even more recently, there's been exclusion of Black, Indigenous, and people of color in clinical trials. But that group of individuals is overrepresented in that 8 to 10% of people who aren't benefiting from modulators because their more rare variants weren't studied. Next. Also, we have to think about consenting people for these very different trials than trials like the modulators. These are early phase, and often first in human means first in CF. So for the modulators, we actually tested those drugs in healthy participants first. But in these therapies, for the genetic therapies, they're usually going to go straight into people with cystic fibrosis. And we need to talk to people about the risks. So redosing may be a problem for some of these vectors. So for example, for AAV, we are all exposed to that virus, and we may already have antibodies before we even get the therapy. And if we develop more antibodies after we get the therapy, that might prevent it from working. Also, if you're in one study, you might not be eligible to participate in the next study, and people need to understand that when they're signing consent. And as Paul said, right now we know that CF is a systemic disorder, so it affects multiple organs. But right now we're really focused on the lungs. And so the other parts of the body that are affected by CF will not be approved or improved by these therapies. Next. So also some considerations by genetic therapy type. So Paul mentioned the different therapies, as did Patrick. So mRNA, the DNA therapies, and something called antisense oligonucleotides, which are just little bits of genetic information that actually don't require a carrier like a vector or a lipid nanoparticle. So if you look at the first column to the right of those different therapies, right now, you can't be on highly effective modulator therapy and be in those trials. That may change in the future, but right now, even if you don't feel like you're getting a great response from your therapy, you aren't eligible to participate in these trials. The dosing schedule on the next column is very different for all of these drugs, or all of these potential therapies. So for mRNA, it's probably going to be daily or every other day because mRNA is very short-lived. For a virus like AAV, we think maybe it'll last 12 months, but we don't yet know. For something that is integrating like lentivirus, perhaps a little bit longer. And then with the ASOs, we probably have to dose people weekly, we think. Also, in the next column, there's a look at theoretical and actual risks. So several of these therapies have the risk of flu-like symptoms, and many of you will have heard that there was a pause in one of the trials because people were experiencing too many flu-like symptoms. So as a clinical researcher, we have to go back and say, what other things can we do to mitigate or make those flu-like symptoms less so that people can tolerate the therapy? So we pause for a little bit because we are trying to make sure that people stay safe during these trials. And then we figure out what we can do to mitigate those problems and then restart the trial. The long-term follow-up for these different therapies is very, very different. So it's mandated by things like the FDA or EMA. So for mRNA, it's only 2 to 5 years, but for lentivirus, it's after now 15 years you have to be followed up, which is intimidating for some people. Also, the last point I made on the previous slide, if you participate in one of these trials, will that prevent you from being in another? Probably not for mRNA because it's very short-lived, but with the viral vectors, it's possible that you may not be able to be in that next trial. With ASOs, we don't think that's going to be a problem. Again, very short-lived. Okay, next slide. So for those of you who aren't familiar with the CF Foundation drug development pipeline, on the y-axis is what phase the studies are in, except for across the top, you'll see the ones that are already approved, and then the different buckets of the types of therapies are along the bottom. Next. So I'm highlighting here the therapies that are focused on correcting the genetics or the protein. So as you heard, we do, in spite of all of the considerations and challenges I mentioned, have 7 therapies, which is really exciting. Next, one that's not quite made it onto the slide yet is the lentiviral trial that's already started in the UK and will be coming hopefully to the US soon. So even though there are lots and lots of challenges, it's okay, Chandra, you can go back though. One more, yeah, thanks. There are lots and lots of challenges. We are at the phase where we are actually trying this again, which I think is incredibly exciting and hopefully makes you all feel excited about it. Okay, next slide. So several people asked in the pre-questions, how do you learn more? Well, there are a number of different ways. So you can go right now to emilys-entourage website, and you heard from Emily earlier about the Clinical Trial Connect, so that'll help match you up based on the answers that you give with potential trials that you could participate in. It also talks about the different trials. Similarly, on the CF Foundation website, there's something called the Clinical Trial Finder, which again describes the different trials, and you can put in different filters. If you only want to hear about genetic therapies, for example, or you only want to hear about infection therapies, you can use that in the Clinical Trial Finder. And then finally, clinicaltrials.gov does actually have information on all the trials that are registered in cystic fibrosis, both here in the United States and some that are happening outside the United States. Okay, next slide. So now I'm going to shift gears just a little bit and talk about something that Chandra mentioned early on, and one of the things that I'm most excited about, and this is in utero therapy. In other words, treating the baby while the mom is still pregnant with the baby when it's been diagnosed with CF. And you'll see that there were 2 talks and a panel, and I'll just briefly mention those. Next slide. So Dr. McKenzie actually was really focusing on and asking the question, why not wait until after birth? Well, we know that we can diagnose babies with CF at birth because their pancreas has already stopped working in most cases. So what's unique about treating a baby while they're still in utero is, one, their immune system isn't as developed. So there's less likely to be a big immune response to the vectors or the way we're getting the information into the cells. Also, they have a very accessible stem cell population, and that's the cells that we really want to get this information into for permanent correction. Also, obviously, this fetus is very small in size, which means you can really get that drug distributed throughout the body of the fetus and at a much lower cost. And then finally, if you need your therapy to get into the brain, there's something called the blood-brain barrier. And in adults, that's very well developed to keep things out of the brain. But in babies, that's not yet developed. So if you need something to get to the brain, then you can do that through fetal therapy. One example of a way that's very non-invasive to the mom who's pregnant is to use the umbilical cord. So this is just an example of them using transfusions, but they've also used that in the case that you heard earlier from Patrick about, where they actually use the umbilical cord to get the drug into the baby. Next slide. Dr. Parenteau also talked about another way that you might actually directly get a therapy into the lungs of the fetus, and that is by injecting it into the amniotic fluid. Of course, babies don't breathe air, but they do have breathing movements, and they inhale the things that are put into the amniotic fluid. So in this mouse model, they were able to show that you could inject a drug into the amniotic fluid. It would go into the baby mouse and then be distributed throughout the fetal lungs, which is very exciting. Next slide. So that's about genetic therapies, but what about the therapies that we already have approved? And so that was the panel discussion I mentioned. So work by Dr. John Engelhardt in the CF ferret model showed that if you actually treat a CF ferret while it's pregnant, the baby ferret will actually have improvements. In this slide, what I'm showing specifically is fecal elastase, which is a measure of how well the pancreas functions. On the top box is G551D and essentially a knockout or nonsense mutation baby who was treated with 770, which is ivacaftor or Kalydeco. And on the bottom panel is a baby ferret who was treated in utero who has 2 copies of G551D, which responds to Kalydeco. And that dotted line actually shows what we think is the normal pancreatic function. So if you only had 1 copy of G551D, most of those baby ferrets didn't actually improve their pancreatic function that much. But if you had 2 responsive mutations, so 2 copies of G551D, almost all those babies who were exposed by the pregnant ferret mom getting the drug had normal pancreatic function after birth. So this is very, very exciting. And we've since seen multiple case series reported of babies being treated whose mom is a carrier by treating the mom. And so you can prevent babies needing meconium ileus or blockage surgery and have their pancreas function be almost normal or normal at birth. Next slide. So those were all case reports that we've been hearing about in multiple case series. What we're doing right now, myself and Dr. Sylvia St. Pettery and Dr. Mike Zaretsky are designing a study to intentionally treat babies who are diagnosed in utero with CFTR modulators to try to prevent complications of CF, and not just their gut, but also hopefully prevent any lung disease, and for boys to rescue their vas deferens so that they will not be infertile when they grow up. Okay, next slide. So just to conclude from my part, we've learned a lot of lessons from previous gene therapy efforts. We really, as investigators, have to make clear the potential risks of trial participation because these are often first-in-human studies. We now have a number of different genetic therapy approaches that are being developed, so lots of shots on goal. There are ongoing and upcoming trials which will increase our understanding of the level and durability of clinical response that may be possible. And finally, in utero therapy may actually be possible for disease prevention. With that, I will turn it back to Chandra.
Thank you so much to our panelists. And now if you would, the three panelists, please come on screen. We will go into our question and answer. If you have participants, like the audience, if you have any questions, please put them in our chat and I will get to that. First, we will start with the questions that were sent in previously. Okay. So the first question, Dr. Taylor-Causer, you stressed on a little bit about prior trial participation and how that could affect someone's ability to enroll. Could you speak a little bit more about current eligibility requirements? For example, how would geographic locations, so if there's a non-US resident living in the US, are they eligible, or what if they are living in Canada? And what about genetic mutations? Like, there's a lot of questions about what genetic mutations are eligible for different genetic therapies.
Those are all really great questions. So I think I'll start with the eligibility for people outside the US. The good news is that when I was first in training many, many years ago, we all across the world didn't work together all that often on clinical trials. But right now, there are multiple different networks, for example, in Europe, in Canada, in Australia, ours here in the US, and we are all talking to each other. And so many times, these studies are being done in multiple different countries. And here in the US, because these trials are complicated to set up and run, there are a limited number of sites. But what we are doing is making sure that we can refer people to the places where we're doing the trials. So many of the people who have participated in genetic therapy trials here at National Jewish in Denver don't actually live in Denver. They live all over the country. So the companies help them get their travel to whichever site is doing the study, and then they're able to participate. In terms of their eligibility based on their previous exposures, part of the issue, and this is a paper that we just actually wrote as an international group and published to talk about that challenge. So, for example, I mentioned that mRNA is very short-lived, and so no one's very concerned that that's going to last for a long time, and if you've given it another trial, that's going to affect those results. Whereas if you get something like lentivirus, people are not only concerned about it lasting a long time, but what if there are later side effects? And so therefore, many people who create the drugs are worried about the liability from the other therapy. And so right now, if you get a genetic therapy, such as a gene therapy, it will often exclude you from being in another study, even though it wouldn't necessarily prevent you from getting another dose of that if we show that it's okay to redose people, for example, with AAV.
So the next question, kind of following up on that, is how do researchers determine which genetic variants will respond to specific therapies? So for example, ENaC or uncommon CFTR mutations, how would the researchers decide what works, what doesn't?
That's a great question. But so for all the therapies that we're talking about right now with the mRNA and the DNA, with the exception of the ASOs that are very specific to a certain mutation, those are what we call mutation agnostic. So they should work for any mutation.
Thank you. The next question is for Paul, and it's about delivery. How are gene therapies delivered? Inhaled, IV, pills, etc. And what are the implications of each of those delivery methods? And if you were interested in a trial, what type of time commitments should working adults expect during trial participation?
Yeah, good questions. The delivery vehicles are varied, as Patrick and Jennifer talked about. So there are 3 different viral vectors that are being tested in clinical trials now: lentivirus, AAV, and herpes simplex virus 1. And then there are more than one flavor of lipid nanoparticle that are being tested. And each of these has unique properties, and we're kind of at a stage where it's very early in using these in clinical trials for lung delivery. So for each example that I mentioned, we're going to have to learn from the clinical trial what the outcomes are, if there are any surprises, if everything goes swimmingly. It's just a matter of enrolling people, going through, in many cases, a dose escalation of the study, and then looking for measurable outcomes that we use in clinical trials. So, I think it's really depends on which therapy. I think Jennifer is better suited than I to address some of the details of what the time commitments are, but there is significant time commitment to participate in a clinical trial, and also time commitment beyond the original just delivery of the materials.
Yeah, I'm happy to jump in there. So agreed. So it depends on what the therapy is. So for example, for the DNA therapies, that's just a one-time dose, but it can take a long time to deliver that depending on how big the dose is. And then you have to do frequent follow-ups at the site to be assessed for safety and whether or not the drug worked. For things like the mRNA trial, you actually have to be on site in most cases to get that therapy every other day if that's how often it's given. So you have to be, for example, moving to your site for a month if the trial lasts for a month. There are some protocols during which you may be able to do some of it at home, but not every institution is willing to let people do that in these early phase studies. So it is a significant time commitment. And again, you're reimbursed for your time. That's standard for that to happen, but that doesn't always measure up against how much time you'd miss from work or school. So it's something that people have to take into consideration.
Jen, I think this is again a question for you. Does gene therapy target all CF affected organs and systems, so lungs, pancreas, liver? And if genes are modified in adults, will improvements occur throughout the body over time?
So that's a great question. And right now in CF, we are only focusing on the lungs. Ultimately, we would hope to be able to affect all of the other all the other organs, but right now, all of them are inhaled therapies. And so they're only going to affect the lungs in both adults and later on in kids if we get them through all these safety trials. But right now we're only testing, I should have pointed out, only testing these out in adults because we want to really make sure they're safe before we move them down into children.
Thank you, Jen. And the next question is for Patrick, kind of along the lines of what Jen was talking about. What is the current status of gene therapy trials for infants and babies? And what are the possibilities for prenatal or in utero gene editing? And what ethical considerations should surround these approaches? It's a pretty big question, but it's something that we've all touched on a little bit.
Yeah, Matt, I've asked the same question of my colleagues, when would you maybe try and do this in younger adults or children? And it's a difficult one because if you don't get a good result in adults, normally you wouldn't progress with the drug down to a younger age. But in CF, it may be that you may get a better result in a teenager or a younger infant, but there's much more risk because we just don't know what's happening. That said, if a lot of these trials prove to be safe, but just not having a great success, there may be some argument for trying that. We have to learn a lot from different animal models. We've got some great data from animal models which say that these therapies could work. We know from other studies from other diseases that if you can treat early, you can get really good results. And I think Jen mentioned a lot of this about, the size of an embryo compared to an adult. You need much fewer cells. The immune system is much less developed at this stage. I think a lot of this is still very much in the basic science arena, and this is talking about treatments which will be developed in the future for people who aren't born yet. But we do need to be very much front and center, which the community is, on people who have CF at the moment. So we can learn a lot from these studies. We can learn a lot from these studies that, adjusting doses, the timing of the intervention, the likelihood of the success or otherwise. But I think, that's for the future, but we can learn from what we're doing on these experiments at the moment.
Chandra, can I just add, to one thing to that, because I want to make sure it's really clear since there's been so much press about these babies that have been treated in utero. The difference is that when Dorothy Anderson described CF back in 1938, it was a fatal disease of childhood, cystic fibrosis. But now we expect, even in the absence of modulators, for people to live into their 40s. And so the reason some of these other therapies have been approved to treat little kids and infants and even in utero is because those diseases that are being treated are very rapidly fatal, which fortunately CF isn't right now. And that's part of the reason there's reluctance to move too fast into kids, because we don't want to cause safety issues for somebody who's expected to live into adulthood without a therapy.
Thank
If
you.
I could just add too, it was a discussion point at the meeting. Historically, the way clinical trials are done with new therapies is they're tested first in adults, and then if safety and hopefully efficacy is proven, then they get tested in younger age people. That's kind of hardwired into the thinking of clinical trial design. But one of the things that was discussed at the meeting is maybe that isn't always appropriate for every problem, as Jennifer just referred to.
Thank you, Paul. We have a lot more questions that have come in, and obviously we can't get to all of them, but I want to get to some of the live questions that have come through. So the first question, what is the realistic best case and worst case timeline for advancing some of these new therapies? Who wants to take a crack at that?
I'll make a start.
Go ahead.
But it's in two parts. So I think if you look at the editing strategies, that there's so much which is advanced there that I think you would have editing strategies which would be ready to deliver in 2-3 years' time. I think the efficiency, low off targets, as safe as we can possibly get it. But maybe this is where Paul was going to come in. It's the delivery. It's the second part of that, which I think is more challenging.
Yeah, I was just going to say that the technologies for doing repair of the gene that were outlined, are advancing in amazing ways. And in clinical studies showing efficacy. So really where our major focus is in CF is how do we get these materials that we know work into the cell types so that they can do their job. And that's where probably our biggest challenge is. And I think, as we've said more than once, from the trials that are ongoing, we're going to learn something new about delivery to the human lung. And anything that works even for delivery of mRNA could apply to other technologies. So we kind of have to let these trials proceed and then learn from them and apply, what works and what doesn't work to build better delivery strategies if necessary, or find one that works great and then start to apply it in more than one way.
I'll be a little more concrete and say, based on the clinical trials, I think that's all very true, but based on the ongoing clinical trials, we will have readouts for some of these trials within the next 12 to 18 months. And that is going to really help us answer that question better and more specifically.
So the next question kind of follows up on that. Thinking ahead, do any of the panelists imagine that CF care centers will need to have specific infrastructure set up to deliver FDA-approved gene therapies locally, such as negative pressure rooms for nebulization, a gene therapy oversight committee, et cetera, et cetera?
That's a really excellent question. And I think it is a possibility, although as I mentioned, for example, for the mRNA trials, there are parts of them right now that if your institution will allow it, those trials can be partially done at home, which means that, for example, you're going to have the right refrigerator, which will have to be part of the drug because these have to be kept very, very cold. So it's possible that some of these therapies will actually be delivered by the patient themselves at home with a lot of extra things in their house, like a -20 or a -80 refrigerator. As far as centers delivering, again, it's a possibility, but we often will probably have more referral basis than we have every single center trying to set up for these trials, because my research coordinator, I don't think is on this call, but she can tell you it is onerous to be able to set up to actually do these trials. And so I'm not sure that every center would have that capability or the funding to do that.
Thank you. And there's a two-part question, or two people asking similar questions, is why do some of the trials go straight into, instead of going into phase 1, they go straight into people with CF instead of healthy non-CF people? And then if these drugs do, have a successful phase 2, is that enough for FDA approval, or do you need a phase 3?
Yeah, questions are excellent. So the reason that these therapies are going straight into people with CF is because healthy people don't have these genetic mutations. And so the regulators and ethical boards don't think it's ethical to give a genetic therapy to somebody who doesn't have a genetic disease. So in terms of whether or not a phase 3 is needed, that's really going to be up to the regulators. Again, you'll hear some of the news about some of these therapies being approved after only small numbers of people have received it, but those are often diseases that are, again, rapidly fatal. Right now, the FDA has indicated that it will still require a larger trial than you would see in a phase 2, which is usually no more than 12 to 20 people in general. So right now, that's where we are, but the FDA is changing, the EMA is looking at these things as well, so it could change in the future.
Thank you so much. We have a ton of other questions. As we're getting to the top of the hour, we can't get to all of them. So, we are going to do a blog post. And so we will try and answer your questions, at that time. So please watch out for that. And finally, we've been, a little bit on EE. We've been really busy here. As you know, we give out a lot of grants. This year, please check out the, the 4 translational grants that we funded. The, and final 2 announcements are forthcoming. There are QR codes here that you can scan to learn more about gene editing technologies from David Liu's lab at the Broad Institute, as well as Dr. Anka Segal's work on phage therapy, targeting a drug-resistant bacteria called Achromobacter. Additionally, I would really like to talk a little bit about the CF Clinical Trial Connect that, Emily touched on earlier. This is a global patient database. It's designed exclusively for individuals who do not currently benefit from CFTR modulators due to either genetic ineligibility, side effects, or suboptimal response. There are companies who are actively recruiting for these trials. So please do sign up and learn about trials and see if they are relevant for you. And you can also find the latest clinical trial information from this website. So I really encourage you to go to our website or scan this QR code to learn more about it. So please do register your children. And we have more than 800 people already signed up on this Clinical Trial Connect. And lastly, we have our gala later this year in October. Please look at this this QR code to find out more about our gala and and the theme of this year's gala is coming into focus. As you can see, there are lots of in the past 12 years, we've gone from basic research to a clinical trial. So we are very hopeful that we will get a treatment or a cure at some point, because we have so many scientists who are spending hours and days and their whole career to try and find therapies for the final 10%. And our work is not complete. We will continue to work tirelessly for those who have CF who do not benefit. Again, the final 10% is who we work tirelessly for. And again, thank you so much to our panelists. We couldn't have done this without you. And lastly, please do not forget about the post-event survey. It's only a few questions, and your feedback is incredibly helpful as we, fine-tune our webinars and bring more of these webinars to you. And finally, thank you again for spending 1 hour of your evening with us, and we look forward to, hosting you again in the near future. Thank you very much.